Bismillahirrahmanirrahim. Dear students, welcome back to my new lecture. Before going to start the new lecture, let me explain the last part of my previous lecture. I think so I couldn't explain it well. The first hormone that was discovered was secretin. There were two English scientists. 1902 में उन्होंने डाक के ड्यूडिनम से कुछ फ्लूइड कलेक्ट किया था और फिर वेरियस बायोकेमिकल प्रोसेस के थ्रू उस फ्लूइड से एक केमिकल को उन्होंने एक्सट्रैक्ट किया था और बाद में उस केमिकल को उन्होंने अनदर डाक के ब्लड में इंजेक्ट किया और थोड़ी देर बाद उन्होंने ऑब्जर्व कर लिया कि उस डाक की जो पेंक्रियाटिक सिक्रीशन था वो इंक्रीज़ हुआ था। इसलिए फिर उन्होंने इस ड्रग को इस केमिकल को नाम दिया सिक्रेटिन बिकॉज इट इंक्रीज द सिक्रीशन ऑफ पेंक्रियास, देयरफॉर दे कॉल्ड देम सिक्रेटिन। और बाद में इसको जो है हार्मोन इसलिए कहा गया क्योंकि वर्ड हार्मोन की मीनिंग है टू ब्रिंग इन मोशन और टू काज एक्साइटमेंट, क्योंकि इस केमिकल ने उसको एक्साइट किया था पेंक्रियास को, जिसके रिजल्ट में पेंक्रियास ने पेंक्रियाटिक जूस की सिक्रेशन इंक्रीज़ की थी। इसलिए फिर जनरल टर्म जो था वो इन केमिकल्स के लिए हार्मोन यूज़ होने लगा। सो let's talk about mechanism of action of hormones. You know that neurotransmitters, hormones, cytokines are also signaling molecule, pharmacological agents such as drugs. All of them show their effects through their specific receptors, and by itself receptors are protein. And you know also that some hormones are lipid soluble, they can cross the cell membrane, such as steroid hormone. Some hormone can also cross the cell membrane because of their small size, such as T3 and T4. And some hormone cannot cross the cell membrane because of their large size, such as peptide and protein hormone. And some also cannot cross the cell membrane because they are highly charged, such as epinephrine and norepinephrine. So first I will show the mechanism of action of protein hormone, that how they initiate a cascade of reaction or how they regulate metabolism inside the cell. So for example, this is one cell and this is its nucleus. I am drawing the nucleus on one side for one specific purpose because I will show here some reactions here. So their receptors are present on the cell membrane and their receptors are called G protein coupled receptors. Why they are called G protein coupled receptor, that I will tell later on. And another characteristic of their receptor is that they possess two domains, one extracellular domain and another intracellular domain. The intracellular domain usually remain inactive until the hormone do not bind with the extracellular domain. And here is one protein inside the cytoplasm. That protein is called G protein. This G protein consists of three subunits: alpha subunit, beta subunit and gamma subunits. With the alpha subunit G, D, P attached. What is GDP? GDP, it is a type of nucleotide. What is nucleotide? Each nucleotide consists of three main components. One is pentose sugar, another is phosphate, the third component is nitrogenous base. If that nitrogenous base is adenine and here is one phosphate, one pentose sugar, then it is called adenosine monophosphate, AMP. If here two phosphate are present, then it is called adenosine diphosphate. If here three phosphate are present, then it is called ATP, adenosine triphosphate. It means ATP is what? It is a nucleotide. And if the nitrogenous here is pentose sugar, one phosphate, and the nitrogenous base is guanine, and phosphate is 1, then it's called guanosine monophosphate, GMP. If here 2 phosphates are present, then it is called guanosine diphosphate. If 3 phosphates are present, then it is called guanosine triphosphate. And usually, if here are cytosine, nitrogenous base, then it will be called, and phosphate is 1, it is called cytidine monophosphate. If 2 phosphates are present, called cytidine diphosphate, and if 3 are present, then it is called cytidine triphosphate. These are all the types of nucleotide. And usually, when nucleotide is in triphosphate state, then it is active, it is energy rich. So GDP is what? It is a type of nucleotide, and usually remain inactive when the alpha unit is attached with GDP. When hormone come and it bind with the extracellular domain, for example, this is insulin hormone. When it bind with its domain, then this domain also become active. When it become active, then it bind with the G protein. When G protein bind here, this is the alpha subunit, beta and gamma. Then this domain, intracellular domain, it remove this GDP from the alpha subunit and attach GTP, guanosine triphosphate. So here is GTP. GTP is attached while GDP is removed. When the alpha subunit attached with GTP, then the alpha subunit detached from the beta and gamma subunits. So beta and gamma came to the ground or came down. This is the beta and this is the gamma. And the alpha subunit move here. This is the alpha subunit with its GTP. Now it is in active state. And here in the cell membrane, one another enzyme is present. This enzyme also have two domain, one extracellular domain and intracellular domain. The extracellular domain is usually inactive site, while the intracellular domain, which is present in the cytoplasm, it is called effective site because it will show its effect here inside the cell. So here is its active site, and here this enzyme is called adenylyl cyclase. It has binding site for alpha subunit. The alpha subunit come and it bind with its binding site present on adenylyl cyclase. So here is now the alpha subunit with GTP. Here is the GTP. When the alpha subunit bind with adenylyl cyclase, it activate adenylyl cyclase. But here, this GTP inside this GTP alpha subunit of G protein, it possesses one enzymatic domain. This enzymatic domain also become active when it bind with its receptor. It then hydrolyze this GTP into GDP. When the GDP convert into GDP, then the alpha subunit detach from its binding site and come down. So now G alpha subunit with GDP is here. This alpha subunit again bind here with beta and gamma subunit. So in this way, G protein with GDP is again produced. Now, what this effective domain does, now it is in active state, it transform ATP, adenosine triphosphate, into cyclic AMP, adenosine monophosphate. This adenosine monophosphate, it in turn activate one another enzyme which is called protein kinase. This kinase in turn, it does phosphorylation of various proteins. For example, I think so in my second lecture I talk about that insulin make permeable the cell membrane of muscle cells and adipose cell for glucose. So If it is insulin, so it will now make permeable the cell membrane for glucose. How it make permeable the cell membrane for glucose? Here are glucose transporter are present inside the cytoplasm. This protein kinase, it start to phosphorylate various proteins which can be in the form of certain transporter for specific materials. So it start phosphorylate these transporter in the form of channel. When these transporter are, when any protein phosphorylate, usually it become active. It then performs specific task. So now they are phosphorylated, they are now in active state. They come and they bind with the cell membrane. So as a result, what happened? Now these are the glucose channel. So if glucose concentration in the blood or outside is high, then through diffusion through this channel, now the channel is, the membrane is permeable for glucose, the glucose then move inside. So in this way, insulin facilitated or insulin made permeable the cell membrane for glucose. And this protein kinase not only phosphorylate transporter protein, it also phosphorylates certain enzymes. So this is enzyme. It is phosphorylated by protein kinase. When this enzyme is phosphorylated, it becomes active. It then take part in metabolic processes. As you know, insulin also help in glycogen synthesis. For example, this is an enzyme which help in glycogen synthesis. It start glycogen synthesis, which is a metabolic process. And it not only phosphorylates enzymes present inside cytoplasm, it also phosphorylates certain transcription factor. For example, this one, transcription factor. Transcription factor. When the transcription factor phosphorylate, it become active, then it start to move inside the nucleus, and you know, inside the nucleus chromosomes are present, and chromosomes consist of genes. This transcription factor then enter into the nucleus and it bind with promoter regions of certain genes, and it then, if this hormone is excitatory in nature, so then these transcription factor stimulate the expression of that gene. If it is inhibitory in nature, then this transcription factor may it inhibit that gene. So here what, when this gene is activated, in response messenger RNA is produced. The messenger RNA leave the nucleus membrane and translate into what? Into protein. And two types of protein are produced. One type of protein, this protein can either be structural protein or they can either be functional protein. Structural proteins are those proteins which take part in the formation of some structure. For example, actin and myosin filament jo ho gaye, they are protein in nature. So structural protein are those proteins which take part in the formation of structures. And functional proteins are enzymes in nature. These are enzymes, and these enzymes then take part in metabolic activities. Is you know that insulin also help in the regulate glycolysis. For example, this can be the enzyme which take part in glycolysis. So as a result, glycolysis will start. So in this way, insulin what? Insulin regulate, when insulin bind with its receptor, it initiate a cascade of reaction. In response, what? In response, the cell membrane become permeable for glucose, and also in response, certain enzymes become active which take part in glycogen synthesis, and certain enzymes are formed which take part in glycolysis. So, as I told in the beginning that hormones regulate metabolism. So I think so आप लोगों ने भी प्रैक्टिकल देख लिया कि कैसे हाउ हार्मोन रेगुलेट द मेटाबॉलिज्म. हमने एक ये फंक्शन भी बताया था, हार्मोन आल्सो रेगुलेट ट्रांसपोर्ट. तो आपने देख लिया कि इंसुलिन ने कैसे जो है इन ग्लूकोज को ट्रांसपोर्ट किया सेल के अंदर. Take the example, for example, if this hormone was let's suppose ADH, अगर ये ADH था तो ADH फॉर एग्जांपल अगर इधर आता तो ये सारा कैस्केड का रिएक्शन इसी तरह होता। और यहाँ पे स्पेसिफिक जो है चैनल्स होते हैं जिसको कहते हैं एक्वापोरिन चैनल। दिस एक्वापोरिन चैनल फॉस्फोराइलेट होते हैं, और जब एक्वापोरिन चैनल फॉस्फोराइलेट हो जाते हैं दे कम एंड attach themselves with the cell membrane, and through it then water move to the outside of the collecting duct cell. कलेक्टिंग डक्ट सेल जो है इम्परमिएबल होते हैं नेचुरली वाटर के लिए। जब एडीएच बाइंड कर जाता है तो एडीएच के रिस्पॉन्स में क्या हो जाता है, सारा कैस्केड ऑफ रिएक्शन हो जाता है। तो इवेंचुअली जो एक्वापोरिन चैनल्स हैं जो सेल साइटोप्लाज्म के अंदर प्रेजेंट होते हैं उनकी फास्फोराइलेशन हो जाती है, प्रोटीन काइनेज उनकी फास्फोराइलेशन कर लेता है, और जब फास्फोराइलेट हो जाते हैं तो दे बिकम एक्टिव एंड अटैच विद द सेल मेम्ब्रेन, एंड इन दिस विद द सेल मेम्ब्रेन बिकम परमिएबल फॉर वाटर। वाटर इज देन रीअब्सॉर्ब्ड फ्रॉम द ल्यूमेन ऑफ कलेक्टिंग डक्ट and enter into the blood. So this was all about that how peptide or protein hormone perform its action. So let me summarize it. This is a receptor for protein hormone. It has two domain: extracellular domain, intracellular domain. Intracellular domain usually remain active until hormone do not bind with it. These receptors are also called G protein coupled receptor because in the cytoplasm one protein is present which is called G protein. This G protein consists of three subunits: alpha, beta and gamma. The alpha unit, with alpha unit GDP is attached. When hormone come and bind with it, then this intracellular domain bind with G protein, and when G protein bind with this domain, then this domain remove the GDP from alpha subunit and attach GTP, or you may say it replaces GDP with GTP. When alpha subunit bind with GTP, then it detach from the beta and gamma subunit. The beta and gamma subunit come down, and this activated alpha subunit together with GTP bind with its binding site present on adenylyl cyclase. As a result, the adenylyl cyclase become active. लेकिन जैसे ही ये अल्फा सब यूनिट बाइंड हो जाती है इस रिसेप्टर के साथ, तो इस अल्फा सब यूनिट के अंदर एक इंट्रिंसिक प्रोटीन एंजाइम प्रेजेंट है। इसको इंट्रिंसिक एंजाइम कहते हैं जो इनसाइड प्रेजेंट है। तो ये एंजाइम पर क्या करता है, ये इस जीडीपी को हाइड्रोलाइज कर लेता है जीडीपी में। तो जैसे ही जीडीपी जो है हाइड्रोलाइज हो जाती है जीडीपी में, तो ये अल्फा सब यूनिट अगेन डिटैच हो जाती है अपने रिसेप्टर से और नीचे आ जाती है। तो दिस alpha subunit together with GTP again bind with beta and gamma subunit, and in this way again G protein together with GDP is produced and waiting for another reaction. Now adenylyl cyclase is in the effective site of adenylyl cyclase is now in active state. It transforms ATP into cyclic AMP. Cyclic AMP stimulate protein kinase. Protein kinase then start phosphorylation of various transporters, various proteins, various transcription factor. ट्रांसपोर्टर जो है वो फिर सेल मेम्ब्रेन में अपने आप को अटैच कर लेते हैं और मेम्ब्रेन के क्रॉस जो है वेरियस मटेरियल को ट्रांसपोर्ट करते हैं, और कुछ जो है एंजाइम की फॉस्फोराइलेशन कर लेते हैं। फिर एंजाइम जो है वेरियस मेटाबॉलिक एक्टिविटीज़ में पार्टिसिपेट करते हैं, और कुछ ट्रांसक्रिप्शन फैक्टर को भी ये स्टिमुलेट कर लेते हैं। ट्रांसक्रिप्शन फैक्टर जो है वो अंदर चले जाते हैं और वो जीन की एक्सप्रेशन को रेगुलेट कर लेते हैं। जीन की एक्सप्रेशन के रिजल्ट में मैसेंजर आरएनए बन जाती है। मैसेंजर आरएनए से जो है प्रोटीन बन जाते हैं, विच कैन बी आइदर स्ट्रक्चरल प्रोटीन और आइदर इट कैन बी फंक्शनल प्रोटीन। सो दिस वाज ऑल अबाउट मैकेनिज्म ऑफ एक्शन ऑफ पेप्टाइड हार्मोन। और अब आप लोगों ने देख लिया कि कैसे जो है हार्मोन जब अपने रिसेप्टर के साथ बाइंड कर जाते हैं तो मेटाबॉलिज्म को कैसे जो है वो रेगुलेट करते हैं, रेट ऑफ केमिकल रिएक्शन को कैसे इंक्रीज या डिक्रीज करते हैं, और सेल मेम्ब्रेन को कैसे परमिएबल करते हैं फॉर ट्रांसपोर्ट ऑफ वेरियस मटेरियल। सो नाउ लेट मी टॉक अबाउट द मैकेनिज्म ऑफ एक्शन ऑफ ऑफ स्टेरॉइड हार्मोन और दोज हार्मोन विच कैन क्रॉस द सेल मेम्ब्रेन। सो लेट्स सपोज दिस वन सेल, one thing it's important to mention here about the mechanism of action of protein hormone. Let's suppose this is the receptor for peptide hormone, and this is its extracellular domain, this intracellular domain. Here is the hormone. This hormone is considered the first messenger because पहला मैसेज इसने लाया था. जो साइक्लिक एमपी था, साइक्लिक एमपी इज कंसिडर्ड द सेकंड मैसेंजर. व्हाट इज कंसिडर्ड द सेकंड मैसेंजर? हार्मोन को जो है, हार्मोन, दिस इज इंपॉर्टेंट पॉइंट फॉर एमसीक्यूज. फर्स्ट मैसेंजर एंड दिस द सेकंड मैसेंजर. फर्स्ट मैसेंजर हार्मोन को, इस हार्मोन को इसलिए फर्स्ट मैसेंजर कहते हैं क्योंकि फर्स्ट मैसेज इसने लाया था. फिर साइक्लिक एमपी ने किया क्या? साइक्लिक एमपी ने इस मैसेज को क्या, कि इस मैसेज को कन्वे किया प्रोटीन काइनेज तक, और प्रोटीन काइनेज ने जो है फिर फॉस्फोराइलेशन कर ली वेरियस प्रोटीन्स की, और जो है वेरियस एक्टिविटीज़ पर जो है परफॉर्म होते हैं. तो इसलिए साइक्लिक एमपी को जो है सेकंड मैसेंजर कहते हैं, क्योंकि साइक्लिक एमपी ने इस मैसेज को क्या कहा, इस मैसेज को रिसीव किया विच वाज ब्रॉट बाय हार्मोन, और फिर इसको ट्रांसफॉर्म किया इनसाइड द न्यूक्लियस, एंड आल्सो इट ट्रांसफॉर्म दैट मैसेज टू वेरियस other protein present in the cell. Therefore it is called second messenger. So this is important, this can be important MCQs कि which of the following is first messenger. आपको जो है हार्मोन दिया जा सकता है ऑप्शन में, आपको जो है ATP का नाम दिया जा सकता है, आपको GDP का नाम दिया जा सकता है, आपको साइक्लिक एमपी का ऑप्शन भी दिया जा सकता है. तो हार्मोन इज द फर्स्ट मैसेंजर एंड which of the following is the second messenger. ये IC MCQs भी आ सकती है. तो आपको हार्मोन का ऑप्शन भी दिया जा सकता है, आपको GDP, GMP etc. So cyclic EMP is the second messenger. Okay. So now let's talk about the mechanism of action of steroid hormone. This is the cell and here is the nucleus. And as you know, steroid hormone can cross the cell membrane. This is the steroid hormone. It crosses the cell membrane, and here is the nucleus, and inside the nucleus chromosome are present, and urease receptor are present inside the nucleus. This is its receptor, and these receptor are in the form of transcription factor. This was the hormone, and when it bind with its receptor in the form of transcription factor, then this receptor transcription factor become active, and it bind with promoter region of certain gene and activate that gene. In response, a protein is produced which can be functional protein or which can be structural protein, and that protein that take part in various metabolic function of the cell. So class finished. In next lecture I will talk inshallah about anatomical relation between hypothalamus and pituitary, how hypothalamus regulate the activity of pituitary, particularly interior pituitary, and then how what are the major functions of hormones of interior pituitary, but in next lecture. Tamat bil khair.